Welcome back to my channel Knowledge Chronicle.
Have you ever wondered, how does your body know when to make insulin? Or how a single fertilized egg becomes a fully grown human with skin, bones, muscles, and brain, all from the same set of genes? The answer lies in one of biology's most brilliant systems, regulation of gene expression. Today on Knowledge Chronicle, we'll uncover how your cells switch genes on and off, when needed and where needed. From E. coli's clever lactose trick to the complexity of human cells, we're diving deep. What is gene expression? First, let's understand what gene expression even means. Your DNA holds instructions for making proteins, tiny molecular machines that run everything in your body. But not all genes are needed all the time. So, gene expression is the process of activating only those genes that are required at a specific time. Think of it like this. Your genome is a huge library, but your cell is only reading the books it needs for the moment. That reading process involves turning DNA into mRNA and then into protein. And the control over which book to read and when. That's the regulation of gene expression. Why regulate gene expression? Imagine if every cell in your body made every protein all the time. It would be chaotic and wasteful. Let's take an example. The enzyme beta-galactosidase in E. coli. This enzyme breaks down lactose into glucose and galactose, which are used as energy sources. But here's the twist. If there's no lactose around, making this enzyme is useless. So, E. coli smartly switches off the gene that makes this enzyme when it doesn't need it. This is a classic example of metabolic regulation based on environmental conditions. Gene regulation also controls development, helping a fertilized egg transform into a full-grown adult with different organs and tissues. Now let's explore how eukaryotic cells, like ours, regulate genes at multiple levels. Transcriptional level, this is the first checkpoint, whether to transcribe a gene into primary mRNA. If not needed, the gene stays silent. RNA processing level. The primary transcript undergoes splicing, removing introns and joining exons. Here, alternate splicing can produce different proteins from the same gene. Transport of mRNA. Even after splicing, the mRNA must exit the nucleus to enter the cytoplasm. This step is regulated to control which mRNAs get translated. Translational level. Finally, even if mRNA reaches the cytoplasm, translation into protein can still be controlled. For example, by regulatory proteins or RNA molecules that block translation. This multi-level control gives eukaryotic cells precision, like a 5-stage security system. Gene regulation in prokaryotes. In prokaryotes like bacteria, gene regulation is mostly done at the transcriptional initiation stage. Key idea: control the binding of RNA polymerase to the DNA promoter. This is achieved by special regulatory proteins. Activators help RNA polymerase bind, turning genes on. Repressors block RNA polymerase, turning genes off. There's also something called the operator, a DNA sequence next to the promoter. It's the binding site for the repressor. If the repressor is bound here, transcription halts. The Lac Operon. Time to meet the star of gene regulation, the Lac Operon in E. coli. Discovered by François Jacob and Jacques Monod, the Lac Operon is a textbook example of how bacteria control genes based on food availability. Here's how it works. Structure of Lac Operon. The I gene makes the repressor protein. Just a quick note, the I comes from inhibitor, not inducer. The Z gene makes beta-galactosidase, which breaks lactose into glucose and galactose. The Y gene makes permease, which lets lactose enter the cell. A gene makes transacetylase, and while its role isn't fully clear, it's definitely a part of lactose metabolism. These three are structural genes, they do the work. Lactose is the substrate, but it also acts as an inducer. When lactose is present, a small amount enters the cell via permease. Some operon is always on at a low level. Inside, lactose gets converted to allolactose. Allolactose binds to the repressor protein. This changes the repressor's shape. It can't bind to the operator anymore. RNA polymerase now moves freely and starts transcription of the LAC genes. In absence of lactose, the repressor stays bound to the operator. RNA polymerase is blocked, no enzyme is made. So, the Lac Operon turns on only when lactose is present, a perfect example of substrate-based gene regulation. We talked about negative regulation, repressor blocks transcription. There's also positive regulation, where a molecule like cap catabolite activator protein enhances RNA polymerase activity, but we'll save that for another video. So, what have we learned today? Gene expression is like a precision machine. Only the right genes are on at the right time. In eukaryotes, regulation happens at many levels: transcription, processing, transport, and translation. In prokaryotes, it's mainly at the transcription level. The Lac Operon is a beautiful example of how a cell reads its environment and decides what genes to express. And here's something to ponder. How long would the Lac Operon stay on in the presence of lactose? The answer: as long as lactose is available and glucose isn't. Once lactose is gone, the repressor takes its place again, and the operon shuts down, like a perfectly choreographed dance. If you found this video helpful and engaging, don't forget, your one like, share, and subscribe will help us create more valuable content. This is Knowledge Chronicle, unlocking the science behind life, one gene at a time.